Mutations are changes in the genetic material of a cell. They affect each cell's function. Mutagens, like X-rays and gamma rays, are carcinogens, which cause mutations that result in cancer. A change in a gene's base pair is called a point mutation, of which there are two kinds. There is one, base pair substitution, and secondly, base pair insertion or deletion. Substitution is when a nucleotide pair is replaced. If the same amino acid is coded, then it is a silent mutation because nothing changes and the same amino acid is produced. Sometimes the mutation will code for another amino acid, in this case leucine, causing a missense mutation. But if it codes for an amino acid to end the protein, it is called a nonsense mutation and results in a dysfunctional protein. These can result in diseases like cystic fibrosis and muscular dystrophy. The second point mutation is base pair insertion or deletion, and that's when nucleotides are either added or lost from DNA. In this instance, a pair is deleted, resulting in a frameshift mutation. These mutations increase chances for cancer. Now, let's talk about what happens when mutations happen in mitosis.
There are two regulatory proteins that control the cell cycle, cyclins and cyclin-dependent kinases. When the cyclin and the cyclin-dependent kinases attach, they produce a cyclin-dependent kinase complex, or MPF, maturation promoting factor. This kicks a cell past the G2 checkpoint and into the M phase, ultimately cell division. When too much cyclin is produced during the interphase and the CDKs attached to them, this causes the cells to constantly go through division, making gene regulation systems to go wrong and cause cancer. As you heard before, point mutations are chemical changes in just one base pair of a gene, specifically the proto-oncogene. Proto-oncogenes are the cellular genes responsible for normal cell growth and division. An oncogene, however, is a cancer-causing gene. Cancer is caused by mutations in the genes that regulate cell growth and division. There are common mutations that are found in human cancers. Mutations on the RAS proto-oncogene and p53 tumor suppressor gene. RAS gene mutation can lead to hyperactive RAS protein and increased cell division. p53 gene mutations prevent suppression of the cell cycle, allowing cells with damaged DNA passing on mutations caused by the damaged DNA to other cells.
In meiosis, mutations occur when non-disjunction occurs, which is when a pair of homogeneous chromosomes don't separate during anaphase. This can cause aneuploidy, a condition resulting in an offspring with an abnormal number of a certain chromosome. Polyploidy is a condition in which an organism has more than two complete sets of chromosomes. A monosomic zygote, which has one copy of a chromosome, and a trisomic zygote, which has three copies of a chromosome, are two different types of polyploidy.